Welcome back to Peking University MOOC Bioinformatics Introduction and Methods. I'm Ge Gao from the Center for Bioinformatics, Peking University. Let's continue our topic. In the last unit, by systematically screening the molecular evolution data and functional genomics data, we constructed a pipeline for screening and selected the DNA de novo methyltransferase DNMT3 as an object for subsequent in-depth analysis. As we can see from the phylogenetic tree, a gene duplication event in a vertebrate generated two copies, DNMT3A and DNMT3B. After that, sequence of two copies differentiated sharply in mammals. In fact, researchers in mammals indicated that there have been functionally significant differences in DNMT3A and DNMT3B. For example, in DNMT3B knockout mice, DNA methylation level is decreased across the whole genome. However, we can observe the similar phenomenon in DNMT3A knockout mice. So, what are the sequence changes leading to such functional differences? By sequence alignment, we can see that there is a DNMT3B specific amino acid mutation in the vicinity of the catalytic center in mammals. It is very interesting that the locus is isoleucine in all DNMT3A and non-mammalian DNMT3B, but turns to asparagine in mammalian. It is very interesting that the locus is isoleucine in all DNMT3A and non-mammalian DNMT3B, but turns to asparagine in mammalian DNMT3B. As can be seen from the structure, there are significant differences in the physical and chemical properties of the two amino acids. In particular, the two amino acids differ significantly in polarity, hydrophobicity, and electrical properties. In fact, the structure analysis indicates that the substitution from isoleucine to asparagine can significantly enhance the binding strength of the enzyme and DNA molecules with negative charge. Based on this, we guess that the substitution from isoleucine to asparagine in mammalian DNMT3B results in the changes in physical and chemical properties. It thus enables the DNMT3B to get stronger methylation activity compared to DNMT3A and non-mammalian DNMT3B. This part of work is mainly done by Li Shen in Professor Gang Pei's research team. Now he is a research fellow in Harvard University. To experimentally verify the above assumption, the direct idea is that for the locus that we constructed two independent mutants and see whether they are consistent with the expected phenotype. However, if we directly conduct this experiment in mammals, it is difficult to exclude the impact of endogenous methylation enzymes. Therefore, we chose the yeast as a model system. As a common laboratory model organism, the yeast is very similar to mammals in epigenetic features. It also has the structure of chromatin. However, there is no endogenous DNA methylation in yeast, thus providing a clean experimental background environment. We first transfer the wild-type DNMT3A and DNMT3B of different species into the yeast. We confirm that mammalian DNMT3B has a stronger methylation activity for chromatin DNA than DNMT3A and non-mammalian DNMT3B. Is this difference related to the substitution from isoleucine to asparagine? To test this, we transferred the previously referred mutant and do a further test. To test this, we transferred the previously referred mutant and did a further test. The results showed that a single amino acid substitution can indeed effectively enhance the methylation activity of DNMT3A, and the reverse substitution will reduce the methylation activity of mammalian DNMT3B. That is to say, the substitution from isoleucine to asparagine contributes to a stronger methylation activity. That is to say, the substitution from isoleucine to asparagine contributes to a stronger methylation activity of mammalian DNMT3B than DNMT3A and non-mammalian DNMT3B. So, is the change caused by the significant differences in the physical and chemical properties of the two amino acids? We further adopted different forms of amino acid substitution to be separately tested. Finally, we found that as long as the substituted amino acids are positively charged or polar amino acids, it will have a similar effect. This confirms the earlier hypothesis. At this point, by means of experimental biology, we've successfully confirmed the two earlier hypotheses proposed by computational analysis. So what's the selective advantage of the functional changes for evolution? Methylation is an important way to suppress transposons and other repetitive elements, and the density of repetitive elements in mammalian genomes has increased significantly compared with the prior. So do the changes in mammalian DNMT3B contribute to its suppression of repetitive elements such as transposons? To test this hypothesis, we further performed experiments in DNMT3A and DNMT3B double knockout mouse embryonic stem cells. We found this locus indeed plays an important role in the methylation of repetitive elements in mammalian DNMT3B. Thus, we further promoted the previous conclusion. Based on the prior learning, we've demonstrated how to apply bioinformatics methods and experimental techniques to address biological problems. In the first class, we have mentioned that bioinformatics is to use calculation methods, develop new technologies, discover new phenomena, summarize the new rules, and finally answer the biological questions. This course aims to lay a good foundation for your future learning and research through the introduction of bioinformatics, basic ideas, methods, and techniques. Here, Professor Li Pingwei and I, as well as the teaching group, sincerely appreciate everyone's support and wish everyone a happy holiday. That's all for the course. Thank you.